Decoding Randomized Controlled Trials, a Biostatistics Perspective **. Welcome to my YouTube channel, where we break down complex medical and research concepts into bite-sized insights. Today, we're diving into randomized controlled trials, or RCTs, from a biostatistics perspective. Let's decode the key elements of RCTs and understand why they are considered the gold standard in clinical research. Randomized controlled trials are carefully designed studies used to evaluate the effectiveness of interventions, such as new medications, therapies, or lifestyle changes. The key here is randomization. Participants are randomly assigned to either the experimental group, receiving the intervention, or the control group, often receiving a placebo. This helps eliminate bias and ensures that differences between groups are purely due to the intervention. Let's break down some critical biostatistical concepts you'll encounter in RCTs. First up, the p-value. It tells us whether the results we observe are likely due to chance. A p-value less than 0.05 is often considered statistically significant. Next is the confidence interval. This provides a range within which we expect the intervention's true effect to lie. A narrow confidence interval means more precise results, while a wider one suggests more variability. Finally, effect size tells us how large or meaningful the difference between groups is. A large effect size means the intervention likely has a significant impact. Randomization is at the heart of RCTs. There are different methods to ensure fair assignment. Simple randomization is like flipping a coin, while block randomization ensures that each group has an equal number of participants at all times. Stratified randomization goes a step further, making sure specific characteristics like age or gender are evenly distributed across the groups. When it comes to analyzing RCT data, we often use statistical tests like the t-test or chi-square test to compare outcomes between groups. But don't forget the importance of the intention-to-treat analysis, which includes all participants, regardless of whether they completed the study. This approach preserves the benefits of randomization and reflects real-world scenarios where not everyone adheres to the treatment plan. Even in RCTs, bias can creep in. Selection bias occurs if the randomization process isn't followed strictly. Performance bias happens when participants or researchers behave differently because they know the group assignments. To minimize these, many RCTs use blinding, keeping participants and sometimes even the researchers unaware of who's in which group. Confounding variables are factors other than the intervention that can influence the outcomes. Proper randomization helps reduce confounding, but it's always a factor to watch out for. To wrap up, randomized controlled trials are a powerful tool in evidence-based medicine, and biostatistics plays a crucial role in designing, analyzing, and interpreting these trials. Understanding the basics of RCTs can help you better evaluate the research you encounter and make more informed health decisions. If you found this video helpful, give it a thumbs up, subscribe to the channel, and let us know in the comments what topics you'd like us to cover next. Thanks for watching, and see you in the next video.